Today we're going to start having a look at a few plant diseases and what plants do to try and defend against these attacks, and moving on to what they can use and what we can use with them, making vaccines. So let's start with having a look at plants. Now, plants get diseases just like, well, people. They're under attack by viruses, by bacteria, by fungi, and of course they can be attacked by insects and well, animals trying to eat them. So some of them accept the fact that they're going to be eaten, and they work on this system to allow themselves to be eaten, to help propagate them having their fruit eaten. Others take a different attitude and try and stop this. Let's have a look at some of these simple diseases and see what we can have a look at. So if we think about one of the most common insect pathogen, that has to be the aphid. Now, what does the aphid do? Well, the aphid, very simply, if we have our plant and we have going along here our phloem, then the aphid simply sticks its mouth part into the phloem and literally doesn't even suck. It gets this pumped into them. Now, of course, if they're doing that, they're gonna basically deprive the plant cells of some of these photosynthetic products. But these aphids can also act as a vector, a means of carrying something, and they can introduce a virus, some sort of bacteria, or some fungi in there. And although these aphids can be destroyed by chemical pesticides, by releasing eating insects such as ladybirds and their larvae, it doesn't sort of work fantastically. There are some other things that are going to get in, and there are some special little worms, and these are called nematode worms. And these nematode worms, they can burrow into the plant, they can feed on the plant roots, and that means the plant couldn't take up necessarily water, mineral ions, and it's going to try and die. So we've got these sort of things, but plants have also got a problem. Where are they? They don't move, and what plants are after are different minerals, and these different minerals may be in abundance or they may not be. One of the most important minerals is magnesium. Now, magnesium's very important because it's a critical part of a chemical called chlorophyll. And we know that chlorophyll is necessary for photosynthesis. If we don't have enough magnesium, then this is called chlorosis. And we see this as a sort of a yellow plant rather than a green one. If we can replace these missing minerals with fertilizers, then the plants normally sort of spring back to life, and they do quite well. How would you check that a plant has got some disease? Well, you'd be looking for certain things. You'd be looking for things like stunted growth. plant's not growing terribly well. And sometimes that can be due to, not exclusively, lack of nitrate, because the nitrate in the soil is necessary to add to the sugars that are made by the plant, and they're turning in those things into amino acids, which will become proteins. We look for things like spots on plants, spots basically on the leaves, and this is usually a good sign of something like black spot. Things like black spot on roses, and these things aren't very nice, and these are typically things like fungi. The big problem with this is trying to eradicate it. We might have things like areas of decay. Just a bit of a plant that's sort of rotting away. Potato blight is one of those. I suppose even the black spot does that on roses. We sometimes see peculiar little growths on a plant, sometimes different colours. Crown galls. This is typical of bacteria. We get things like malformed stems. And these are often due to things like aphids, or the ones we can't see, these nematodes. We get this discoloration. Chlorosis, typically mineral deficiency. And the one that most people seem to notice, we get things like bent leaves, caused by sort of perhaps mites and things like that. Aphids, you see the leaves being chewed. So we've got real visible signs, and these could be things like there. Caterpillars, aphids, mites, all sorts of different things going there. Identifying what's going on is usually easy. We just look at the plant, we can say, yeah, there's something wrong. Identifying what the actual disease is not so easy. Many diseases give similar symptoms, and so trying to sort of work out which one of these is causing the problem makes it harder to treat plants. But plants do have a whole means of trying to defend themselves, and they go about it in lots of different ways. First of all, they've got physical barriers they can put in. So they've got strong walls anyway, which hopefully should try and resist many things getting in there. Some of them have these waxy cuticles. We've got things like bark on the tree, which, you know, is particularly difficult to get through. Plants can get quite crafty. We can actually get leaf fall. Something's invading my leaf, right? I'll just drop it off. And those leaves could be lost. So we've got these physical barriers. We've also got various chemical barriers, and many plants produce wonderful sort of defences. One of those that you are probably aware of is mint. Mint tastes nice, but it's also used as a sort of a mild antiseptic. Another one that you might meet is witch hazel. Sometimes put on wounds, and these are mild antiseptics and they're used in sort of some medicines. If you don't want to, however, just get this working against minor little things, you can go for bigger chemicals that can cause lots of interesting problems, and one of those is the foxglove. The foxglove gives out a chemical called digitalis, and digitalis is particularly good if you want to try and kill someone. It's actually used as a heart medicine, but in quantity it can actually cause the heart to stop. So plants learn to sort of not eat things like this. Some plants put physical barriers to try and help this as well. Things like cacti, gorse, thorns, trying to stop them getting in, which are another physical barrier. Some plants go for sort of hairy stems to try and sort of make life more difficult, and some use poisons like the nettle. And I don't know whether to put the nettle as a physical barrier or a chemical, because it's really both, because we've got the poison in the nettle here as well. So physical barrier is something like the sting of a nettle. So we've got a fair number of systems used here to try and prevent attack. One of the most interesting, which is neither of those two, is simply the use of mimicry. And here some plants droop to mimic that they're being unhealthy, and an animal comes along and says, oh, I'm not going to eat that, that's not very good. Some make it look like they've got butterfly eggs on them, and if they've got butterfly eggs on them, well, the butterflies will say, oh, I'm not going to go and lay my eggs there because they've already got caterpillars on them, so what I'll do is I'll lay my eggs somewhere else. And of course the plant has escaped. Now, we've used plants to try and help us for literally centuries, but it's not usually enough. And over the last few hundred years, what we've worked on developing is something called a vaccine. Moment we're suffering from COVID-19 throughout the world, and our hope relies on a vaccine. A vaccine's a chemical that we can sort of inject into us so that we can basically help identify COVID-19 when the coronavirus gets into us, and it can try and take out the coronavirus before it gets too serious. There's a lot of controversy on vaccines. Why? Well, I don't know really, because generally vaccines are quite good, but there are some people who are convinced that they are evil and they're going to try and destroy them. Yet often these vaccines that are curing diseases aren't that harmful. They're not very harmful at all, and the diseases can be terrible, but people say, oh no, I mustn't have that vaccine. Why not? Oh well, a friend of a friend's friends said that, and so we don't take the vaccine, and then people die. So vaccines seem to be quite good. Let's have a look and see what a vaccine is and how it actually protects. So vaccines were developed by a guy called Edward Jenner. He wasn't probably the first, but this was the first one using the idea of cowpox and eradicating smallpox. So how does it work? Well, basically some pathogens can really make you seriously ill very quickly, and you die before your body manages to make the right antibodies. Right, so we've got the disease, and it acts quickly. Basically, it acts so quickly you haven't got time to defend yourself, so you get little response, and usually you die. So the idea behind a vaccine, which is sometimes called immunization, is that you are given either dead or inactive parts of a virus, or maybe a bacterium, or maybe something else, and this basically stimulates the white blood cells to produce antibodies. Now, some people say they're given a little bit of this and they really do get the disease. It's usually not true, because they normally only get it for these symptoms for perhaps a few hours, and people complain, oh, I've got this in my arm, oh, it hurts, and I can't cope with this. And quite often that's more psychological than anything else. Occasionally something does go wrong, but it's usually fairly safe. So we've got antibodies, and these go into special cells called memory cells. These are a special type of white blood cell. If your body then meets the same but live pathogen that has this, then the memory cells get and start stimulating the production of antibodies quickly. And that means that the disease doesn't get such a chance to act quickly, and there is a large immune response rather than a little one, and death doesn't occur. So this is why we've got this idea of trying to protect people from this coronavirus. So let's see what's happening with this coronavirus. So I've got my coronavirus, and it's got its little bits and pieces that we can use to identify it. Now, if I can take one of those off, and I can now basically put it into some creature's body, then it is possible that we can make an antibody to this. Now, this takes this magic word called time. So we want to speed up this time. So if instead of giving the whole virus, we basically take and inject into someone just that little bit, then your white blood cells, they're going to sort of recognize this as a foreign object. They will then process it, and in time they will make an antibody to this funny thing that they've got put in them. So now we've got a population with these things in them. If they get the coronavirus, and they probably will, and it could happen many times, then this antibody is produced quickly, and basically here you've got your coronavirus is basically stopped dead in its tracks. People think that they won't get something like coronavirus again, and this is not true. Basically, if coronavirus is out in the population, then people are going to get it again and again and again. And this is the role of the antibodies. So let's suppose we have a patient, and this patient doesn't have the vaccine, and they get this coronavirus. Now, unfortunately, this person is going to be ill, but let's suppose all goes well, and now here they recover. Now what they should have in them, and this is the hope, they should have antibodies to this funny little thing inside them, so that if at a later stage this person again picks up this coronavirus, nothing happens. Basically, they do get an infection, it does get in, but the antibodies destroy it, and the person here, well, they're happy because they stay well. What they're doing with some people at the moment is they are basically extracting here any antibodies that they've got. So we take out those antibodies, and we could put them into someone who is ill, who perhaps hasn't made sufficient antibodies yet, and this will stimulate their body to start producing these antibodies. So this is a race. It's a race between, will the disease kill you first, or will you manage to produce enough antibodies to take out the disease and stop it? Now, doctors use vaccines to protect us against bacterial diseases such as tetanus, diphtheria, and viral ones such as polio, measles, mumps. There is the famous MMR vaccine, which protects you against measles, mumps and rubella, and they save millions of lives. Just by taking some bit off of this virus. But there are problems. The virus can mutate. There are problems that this antibody in the body doesn't last very long. And for coronavirus, we don't know. So they are trying to extract this bit of the virus. They are trying to get this things here, trying to sort of develop a suitable antibody. If we look at one of these coronaviruses, it has on its surface perhaps several different proteins, various different things to identify it. Choosing the right one could be important. If this virus mutates, and it has different shapes, then they won't be able to be protecting the body. So this vaccine, if they chose to defend against this one, it will work on this one and this one. If they chose this one, it would work on this variety, but not on that variety. It's just a different shape. So if we draw this up, and I've drawn this once before, but I'll do it, it's worth doing again. We have our antibody. Our antibody is typically a sort of a Y-shaped molecule, much like this. And this antibody can stick to perhaps part of the virus. And with this antibody on here, then simply the virus can't work. So we've got these antibodies, and the virus here with this thing on there, I'm going to call that an antigen. So this in theory makes you immune, because you get this virus with all these bits stuck on and the virus is disabled. So it is a case of trying to put a small amount of this stuff, but it's choosing the right amount of the stuff and making sure we've only got that stuff, not full viruses. Now, this is where something called herd immunity comes in. Herd immunity works if we've got, let's say, a fair number of the population which is immune. So let's suppose we've got, I don't know, let's say 80% of the population has got immunity. Now, what we have at the moment in this country is about 10% immunity. We're guessing at that. But if I do that, then for every 10 people that I've got, then this person and this person could get the virus. So let's have some people here. So these people here are our 80% immune, and only these people could get the virus. And let's suppose one of these people does get the virus, like that one. They could only pass it on to that person, and that person would possibly never see those two, and so this disease is not being effectively passed on, and this is going to have an R of basically less than 1, and the system's going to die out. I want looking again at the screen. Let's look at only having 10% of people immune, and if we've got just 10% immune, then you can see that all these people here could get the virus. So if this person gets the virus, then easily they could spread it onto here, which could give me an R of probably much greater than 1. So how can we help this? Well, let's look at, rather than everyone getting the disease, we could put a vaccine in place. So let's look at what happens if we've got a vaccine. Let's take our 10% of our population with the disease. So that's 10% who've had the disease. They've got, in fact, hopefully some immunity, but we can now manage to give 80% of the population a vaccine. So let's give 80% of the population a vaccine. Some may have had the virus, and so we're left now with only some people who are able to get the virus. And so if one of them catches the virus, now, much like having this large herd immunity, we've simulated that, and we've got no spread. And with no spread of the virus, then we're looking at things looking a lot better. But unfortunately, we're not at this stage yet with the coronavirus. We don't have an effective vaccine. And people say, well, why can't we do this? Why can't we get this vaccine in place? Why can't we test? Why can't we do this? Well, you've got to create the test first. We've got to create the vaccine. Viruses are incredibly small, and if they're incredibly small, we can't actually see the thing that we're actually trying to get off. We're trying to get this protein off, but how do we know? How do we know we've got the right bit? How do we know we haven't got any virus in there? It does take time, it does take effort. It's not as simple as, oh, let's get some virus, right, okay, let's chop it off, right, that's 10 minutes later, okay, let's now take some of that let's inject it into a person. Yeah, okay, they're all okay. Let's take their antibodies out. Yep, that's another day gone. Yep, and now we've got our effective vaccine. It doesn't happen like that. It might happen like that in Hollywood, but in the real world, vaccines take a while to develop. There are quite a few diseases that we don't have vaccines to. HIV's been around for quite a few years, and we don't have a vaccine against that. Why not? Because at the moment there are still great challenges. It's too difficult for us to do it. At the moment we're trying, but we're not succeeding. We can make vaccines, but they don't always work. And once you've got a vaccine, this chemical, we've usually got to put it into something. So we normally put it into some sort of carrier that we can sort of then inject into a body. And some people, they do react to that. But they are the sort of 1 in a million, 1 in a billion. And if 1 in a million is going to react badly to this vaccine, or even 1 in a billion, should we all stop taking it? Thinking, oh yeah, I might be that 1 in a billion, when we know that some diseases are going to have a very high mortality rate. Coronavirus, fortunately, isn't that dangerous. It doesn't kill everybody. If this had been a disease like Ebola getting out, then the system could have been a lot worse, because more people would be dying, more people would be catching this infection, and we'd have a problem trying to sort of contain it. The only way of containing at the moment with no vaccine is social distancing. Now, how does social distancing work? And there's a lot of talk about this, so let's spend just a moment on this. I've got 2 people. This person, unfortunately, has the infection, but they don't necessarily know about it. They might sneeze, they might cough, but if somebody is 2 meters away, then they're probably unlikely to get it, even if they pass this person by. Whereas, if we make the social distancing smaller, when this person with their infection passes it on, then the likelihood here, well, it's probably going to double, because the further I can get people away, for every metre, sort of halving. But it's still only a likelihood. If we bring things down to much less than 1 metre, then the certainty of both these getting us is going to be much higher. I watch the news like everybody else. I watch people walking around, and I worry how close people get. Now, if 2 people walk past each other, this one's infected, this one isn't, and even if they do it at a close distance, the chances aren't basically that high. If you stay in contact with that person, yes, your chances are going up. But that's how this virus spread in the first place. This virus spread by close proximity. This virus spread by contact. So we have a problem. We have to remember that when we come to try and cure a disease, the virus, there is basically no cure. Once you have the virus, it's up to you to try and do it. If we've got some bacteria, then we do have for these some antimicrobials, antibiotics, and they work. But sometimes people say, oh, I'm not going to have an antibiotic. What they have instead, they go for something else, and they basically go for something like ibuprofen or paracetamol. They go for these painkillers, and these simply stop the symptoms. They don't stop the bacteria. So we can stop most bacteria with antibiotics, but for viruses we can't. We can't stop a virus with an antibiotic. The only thing that we've got to try and stop these is an antibody, and this comes with vaccination. And until we've got vaccination, then our only defence is social distancing. Otherwise, with only 10% of the population immune, then these other people become infected, and then these people become infected, and we see this racing through the population. Social distancing is important. Washing hands, yep, that's a good way. That's really effective. But the best way, and the only way that governments so far have managed to succeed in defeating all of these problems, has been simply using a lockdown. This is extreme social distancing. The lockdown's effective because people don't meet each other. If we are now out in the wide world, then keeping our distance is the most effective way. Get closer and closer, the more likelihood of the disease being transferred. It's a sobering thought, isn't it, that the thing that we want as humans, we want to get out, we want to meet other people, we want to be with people, we're social animals. Yet that very thing is what the coronavirus capitalises on. We've been apart, we've been succeeding really with lockdown. Most countries are getting things under control with a lockdown. New Zealand did fantastically. Early lockdown eradicated the disease from the country. As soon as they let anyone in, it sort of returns, and then they've got a big problem. If people return, if people have got this thing, it can start up again, the so-called second spike. And sometimes these second spikes are worse than the first spike, because people say, oh, I've done that before, oh no, I'm not going to do that again, no, it won't happen to me. And of course it does. So vaccines there to try and help us. They might work, they might not. Our hope is that they do. Otherwise, these sort of social distancing measures may have to stay with us for a very, very long time. With that not so nice thought, if you found this useful, then please subscribe, and I'll see you next week when we'll do some more GCSE biology topic by topic. Until then, stay safe. Goodbye.